everyone. I'm Samantha. I'm one of the engagement coordinators with Saliva Direct, and along with Brittany Choate, we're going to be moderating this quarterly salivary diagnostics webinar. We have three exciting, distinguished guests here that are going to present on their research. We have Dr. Luke Davis from Yale.
Thank you so much to Brittany and Samantha for organizing the webinar today. It's really my pleasure to talk with you about my work entitled Feasibility and Sensitivity of Saliva Direct GeneXpert MT-RIF Ultra for TB Diagnosis in Adults in Uganda. And this work is work that I've carried out in collaboration with the Infectious Diseases Research Collaboration based in Uganda and Dr. William Rawodria. I think tonight my main collaborator, Mr. Patrick Byanyima, is in the audience and joining, and we carried out this work together. I am a physician and epidemiologist based at the Yale School of Public Health, and my focus is on TB diagnostics in high HIV prevalence settings, in particular studies that promote implementation of these technologies. Just a little bit of background for the audience who may or may not be familiar with tuberculosis. Tuberculosis is one of the top infectious killers in the world. Really only COVID-19 is ahead of that. Given the decline in the pandemic over the last couple of years, I think in 2022, tuberculosis likely unfortunately will remain the leading infectious cause of death. About 1.6 million people died in 2021, and it's the leading cause of death among people living with HIV. One of the main reasons for this high morbidity and mortality is that a large proportion of individuals with TB do not get diagnosed. At the right side of the slide, you can see of the 10.6 million people that are estimated to have TB in 2021, only 6.4 million are known to be diagnosed and started on treatment, leaving over 4 million people around the world who either had symptoms but were not successfully diagnosed or treated, or may not have had symptoms and may have been unaware of their condition. So given this challenge, we have to think a little bit about what's the optimal strategy for diagnosing TB. And I think that's one thing that has changed, even though we have this large proportion of people that haven't been diagnosed, is our paradigm for understanding where we should diagnose them. And so if you look at this slide, this is from a recent review article by Delia Galletti and colleagues from 2022, where she's talking about a different paradigm of TB. Traditionally, we focused on people with active tuberculosis, shown over here at the right-hand side of the slide, this 6 million or so that I showed in the previous slide. These individuals have microbiological positive tests for TB, and they can transmit to others. Often they have large numbers of bacilli that are visible in their sputum when it's examined under smear microscopy, which is the most common test for TB in the world, or in culture, which is the most sensitive test. But we now know that there's a significant proportion of people from this review of prevalence surveys, about 12 prevalence surveys done over the last decade in Africa, show that about 44% of all people with active TB have what we're now calling subclinical TB. So these individuals don't report any symptoms, and they're only identified through the use of chest radiography, which identifies abnormal lesions within their lung. Many times these individuals are culture positive, and therefore we believe that they could spread TB to other people. In addition, there's earlier phases of TB, and I can just say that about 2.5 billion people in the world have latent TB infection, are at risk for progressing, and every year we know we're getting more people with incipient TB that will likely progress in the subsequent 2 years. And gene expression profiling studies, as highlighted by Zach and colleagues, kind of highlight that this phenotype slowly develops. And so we need to be thinking earlier about how to identify these individuals. So in terms of TB diagnostics, today's tests, really, as I mentioned earlier, are primarily mycobacterial culture, shown at the bottom portion of the slide. There's two forms, liquid culture and solid culture. And I'm showing you an image here of the mycobacterial growth indicator tube, which is the most sensitive test that can be done. It can detect about 1 colony forming unit per mil. But culture is really not widely available. And in the last 10 years or so, nucleic acid amplification technologies have really come to prime time. In particular, modules such as the GeneXpert MTB-RIF assay, which are automated assays that have self-contained amplification and have been able to reduce the problems with cross-contamination that were a limitation previously in scaling out TB. Now, these tests aren't quite as sensitive as culture, but they have a limit of detection around 10 colony forming units per mil. And depending on the flavor of tuberculosis, individuals that have what we call smear positive disease with large amount of disease, about 98% of those can be detected, and even smear negative individuals, around 65% of them can be identified. But unfortunately, these tests are difficult to implement because they all require sputum, and patients often are not able to produce sputum. Sputum is one of the ways it requires patients to cough, which is difficult for young children or people that don't produce sputum. And in addition to that, there is a risk of infection from transmission of TB from person to person. And in 2014, WHO convened a meeting to establish target product profiles for so-called non-sputum tests. And the goal is really for these non-sputum tests to match the performance of nucleic acid amplification testing, or NAT, on sputum, but without the need for sputum. These would have minimal sample prep and potentially could get turnaround time with under an hour at a low cost. So why not saliva? We're here at the Saliva Detect webinar, and it turns out that there's a real bias against saliva in the TB world, going back to the performance of saliva in culture-based regimens theoretically. So laboratory guidelines put out by the Global Laboratory Initiative, shown here at the left, often show salivary here as a suboptimal type of sample. But a recent systematic review done in 2015 really found that there were no studies that assessed the relationship between sputum quality and the yield of PCR-based tests. So before we got involved in our direct saliva diagnostics research, we did a proof of concept study where we looked at the cross-sectional performance of GeneXpert technology that I showed in a couple of slides back among 1,782 individuals undergoing evaluation for tuberculosis. And we stratified these individuals into 4 groups based on the appearance of their sputum, as shown in the previous slide. And I'll just have you focus on this top line, which is the sensitivity. And what you can see is that the highest sensitivity was actually in the subgroup of individuals who are characterized as having salivary sputum. You know, why would that be? Well, it may be easier to get nucleic acid DNA, the targets for the MTB-RIF assay, out of saliva than it is out of sputum. And there may be potentially fewer inhibitors as well. And so we found this to be quite promising to move forward with this study that I'm going to be talking about today, which was recently published in Microbiology Spectrum, which looked at the feasibility and sensitivity of saliva combined with this GeneXpert assay for TB diagnosis in adults in Uganda. So here's our research question, I think, which was well captured in the previous slide. But because this was a preliminary study, we wanted to focus on individuals who we knew were sputum expert ultra positive. We did this because this was a small pilot study, and of the population that was being studied, only a small proportion would actually have TB. So we focused in on those individuals in order to be able to determine the sensitivity of the expert saliva, although we knew we would not be able to look at the specificity. Our work is done in Uganda at the China-Uganda Friendship Hospital in the Nuguru district of Kampala. And Uganda is a high HIV/TB burden country, a population of about 46 million, located on the north of Lake Victoria in sub-Saharan Africa, in East Africa. It has a prevalence around 200 per 100,000 in terms of TB. About 7% of the general population have HIV, and about 30 to 40% of individuals with TB also have HIV. And so we carried out this cross-sectional diagnostic study. You're seeing here a picture of a representative waiting room at one of the clinics where we work. They were there for inpatient or outpatient TB evaluation, and we enrolled consecutive adults age 15 and above who had cough of any duration up to 6 months. These are individuals thought to be at high risk for tuberculosis in this setting. We also limited to individuals who had the spot positive sputum expert ultra assay. They got symptom screening, HIV ELISA testing, and CD4 positive T cell count. And we looked at the performance in reference to 2 possible reference standards. One was the mycobacterial culture, which is the ultimate standard for most studies in TB, 2 samples, 1 solid, 1 liquid culture on sputum. And then we also looked at a second reference standard, which was the expert ultra on sputum, because this really is the de jure or de facto standard that you find in most low and middle income countries. And we did the saliva expert assay, collecting at least 1 ml of saliva from each individual, and then diluting that 1-to-1 in a sample reagent, which is a processing reagent which consists of isopropanol and sodium hydroxide. Its purpose really is to decontaminate the specimen of mycobacteria and also to help break down any mucoid elements in there, such as are found in sputum, to help bring out the DNA. Our two outcomes were feasibility, defined as the proportion who actually had Saliva Expert Ultra successfully reported. And then we had two measures of sensitivity in reference to the two reference standards that I mentioned earlier, the proportion that were Saliva Expert positive out of those with sputum culture positive, and then a similar proportion out of those with sputum expert positive. So our first result was that we found that out of the individuals that were enrolled in the study, about 98 patients were eligible for the study. Only 16 were unable to produce saliva or did not provide saliva. And largely this was individuals who left the waiting area prior to produce. It wasn't that they were unwilling or unable to produce saliva. And so we had 82 patients who had sputum expert ultra positivity. One we excluded because of indeterminate culture result, leaving 81 for the primary analysis based on sputum culture. And then you can see the breakdown here of the positivity in terms of saliva expert results. And in the next slide, I'll actually show the sensitivity. But first, let me just tell you a little bit about these participants. They were, median age was around 30. This is typical of the population there. Men were the predominant group who were identified in this study. 91% of them were receiving evaluation as an outpatient. About 22% were people living with HIV with a wide range of CD4 counts, but generally quite low. And a very small proportion, only 40%, were on antiretroviral therapy. In terms of the cough, everyone had cough, and about 21% had cough of more than 14 days, and about 84% were sputum AFB positive. Here, what we're showing is a comparison of the amount of MTB DNA as picked up by the Expert Ultra assay, comparing saliva and sputum. And what you're seeing here is, if you look along the axis, the diagonal axis here, there's a lot more shading in the lower area, suggesting that the semi-quantitative measure of MTB DNA as measured by the Expert MTB-RIF assay is higher in sputum than in saliva. So that's not surprising. Sputum is coming from the lower respiratory tract where TB lives. Saliva is coming from the mouth. And about 80% or so of the sputum expert samples were either moderate to high, whereas only around 20% of the saliva expert assays were moderate to high. Here are our results in terms of sensitivity. At the top line, you can see in reference to sputum culture, Saliva Expert detected 89% of all the sputum culture positive with a 95% confidence interval to 81 to 95%. And there was a big difference in terms of whether the individuals were sputum AFB positive, which is thought to be the individuals that are most likely to transmit TB, have the highest burden of TB. About 96% of the time, it detected that. Only 50% in individuals who are sputum AFB negative. And there was a similar gap between people without HIV who were thought to have a higher burden of bacteria or mycobacteria in their lungs than people living without HIV who become sick at a lower level of mycobacteria. And the sensitivity in them was only 71%. And interestingly, there really was no difference whether we looked at the sensitivity in reference to sputum expert or sputum culture, 89% sensitivity. So some of the limitations of the study is that the small population, a small cohort, there was a high proportion that were AFB positive, which may make the Saliva Expert look better than it would with a lower proportion AFB positive, a more typical proportion. We did not enroll children, and we did not have individuals with a non-productive cough, which are the ones who this assay would be most helpful with, to be quite honest. And also we used this case-only design, which can have some limitations, but when we went back and looked carefully, we really didn't identify any individuals that were excluded that would have been likely to change the sensitivity of the result. And that's kind of described in some detail in the paper. A second limitation is that the sample collection and yield approaches, there's some unanswered questions. We collected the saliva after patients had coughed up sputum, and we're not sure if that might have evacuated the mouth of some of the bacilli and therefore lowered the sensitivity, or if potentially having them cough might bring up more bacteria from the lung, and there might be an important aspect of collecting saliva from patients. Of course, if that's the case, then that might reduce some of the benefits that we'd like to see in terms of decreasing infection risk by not having patients cough with saliva. And then finally, there's some important questions about aerosolization when processing of saliva. There have been studies that have been done showing that sputum, when it's processed on a desktop, as might happen in low and middle income countries where often a biosafety cabinet is not available, it can be aerosolized. And so the sample reagent ratio between the amount of sample reagent that you add to the sputum has been shown at a ratio of 2 to 1 to decrease that infection and aerosol risk. And so we don't know if that's the case with saliva with a 1-to-1 dilution, and we'd have to look at that trade-off because a higher dilution might also reduce the overall sensitivity. Our future directions are we recently received an excellent score on a proposal to look at this in a different population, a community-based population in Colombia, looking at outpatient clinic and also in a high TB contact investigation. This is in partnership with the Centro Internacional de Entrenamiento en Investigaciones Médicas, or CIDAIM. And we're also proposing to look at not only saliva, but also oral swab analysis. I really want to acknowledge the whole team that helped carry out this work. In particular, I'd like to acknowledge Patrick Byanyima, who did all the sample collection, recruitment, and patient processing with help from the other individuals on this slide, and Amanda Gupta, who helped with the analysis. And I definitely want to thank my colleagues, Dr. William Wadra and Lawrence Wong, who are the leaders of this particular study. We also want to thank our funders at NIH, the D43, which funded Patrick's work, and then the grants, other grants listed there. This is our summary slide. We found that saliva was highly feasible and sensitive for TB diagnosis using NAT, and meets the WHO target product profile for a non-sputum test that was referred to early in the background to this. And our future studies will need to try to look at potentially enhancing sensitivity in individuals with polybacterial disease, measuring specificity to see if it is indeed high, as it's been found for sputum, looking at sputum scarce populations, and then also addressing these infection control risks. So for that, I'll stop and see if there's any questions. Thank you.
Thank you so much. If anybody has any questions, feel free to either type in the chat, or you can use the raise your hand feature, and Brittany or I can go ahead and unmute you.
I'm curious, just to start things off, if there are, or what the next steps are or plans are for taking this information and moving it forward, either expanding the population or with publications or what have you for advancing this and using it as a next step.
Yeah, thanks so much. I mean, I think really it's getting the test closer to the population that needs it. We started out in a clinic-based population, and all of whom were able to produce sputum because we wanted to just see as a proof of principle whether saliva has enough bacilli to detect TB in individuals like this. And I think the answer to that question based on this study is a resounding yes. We like to do some laboratory experiments to understand some of the biosafety issues, also some spiking experiments to kind of understand a little bit more what might be the true limit of detection in saliva. And then if we're successful with some of those, I think getting it out into the community, we have a real interest in active case finding. Early in my talk, I highlighted the importance of finding those folks who don't have symptoms around the community. And one important way of doing that is in the context of contact tracing. And so we think that saliva could play a powerful role there in terms of simplifying collecting sputum in the community, decreasing a lot of the stigma that might be associated with producing sputum, and just simplifying it from collecting from some of the key populations who are at risk, like young children who have trouble coughing up sputum.
Thank you, appreciate it. And I see that we have a note that came through in the chat from Shyam. Do you have a sense of the CT values for positive saliva sputum specimens?
Yeah, it's a good question. And we weren't able to extract that information from the GeneXpert machines in the way that we did the current study. We had the semi-quantitative results. And so I think we could probably back calculate what the CT values are. But in the next study, we're going to be very rigorous about recording the CT values from both sputum and saliva, and from the swabs that we'll be doing to kind of really understand what are the differences between those. And I think that'll give us more insights into how we actually have to collect the samples in terms of the order of collection, whether patients should cough or not, and how collecting multiple samples might impact the overall kind of limit of detection of these different assays. So great question, Sham.
Well, if anybody has any further questions, we will have time at the end of all three presentations for additional Q&A. So thank you so much, Dr. Davis. And now we will be moving on to Dr. Paul Dastoor from the University of Newcastle, who's going to share some research on saliva testing for diabetes.
Good afternoon, everyone. Hopefully you can all see that. All right, I'll kick off. My name's Paul Dastoor. I'm Professor of Physics at the University of Newcastle here in Australia. So for most of you, I'm tomorrow and it's in the morning, so I am your future. And what I'd like to do today is, first of all, thank the organisers, Brittany and Samantha, for inviting me to come and talk to you today. It's lovely to have that opportunity. And also to thank Dr. Davis for a very interesting talk. I really enjoyed that talk previously, so I don't know if I'll be able to follow that at all. So I'm going to talk to you about our work here at the Centre for Organic Electronics on using semiconducting inks, on using electronic inks to build sensors, specifically for sort of point of care applications. So what are these electronic inks? So I have been working in the area of organic electronics for nearly 30 years. And these are materials that are polymers or plastics that can act as semiconductors, which is really quite remarkable. Most of us associate plastics with being insulators. We make electrical sockets out of them. We make plugs out of them for the reason that they don't conduct electricity. But actually, it turns out there is a special group of plastics or polymers that do actually conduct. And more importantly, there are another group that actually act as semiconductors. And once I have that, I can make electronic devices, just like I can make the electronic device you're used to in your phone or your computer. Those electronic devices are all based on silicon, which is hard and rigid. In this case, what I'm talking about are a set of materials that I can dissolve up in solution. And I've shown a picture here on the left. So we have inks. Now, the sorts of things that we can make are things like solar cells, and we've done an awful lot of work in terms of developing printed solar as a technology, and that's another area that I won't talk about today. But today I'm going to talk about what work we've been doing on building transistors. And the exciting thing about these materials is that, first of all, the fact that they are inks means that we can print them. We can manufacture in a completely different way. And the sort of printing that we're talking about is roll-to-roll printing, not 3D printing. We're talking about the simple printing that you use to make newspapers or wine labels. But now we can do that and make electronic devices at massive scale. And the other feature of these materials is that they're all based on carbon. These polymers are all carbon-based. And so what that means is that it's actually quite straightforward to embed biomolecules into them and to use those biomolecules as the moieties that do sensing. So the center that I head up here at the University of Newcastle has been going since 2007. And it's about 30 people, about 15 postdocs, and about 10 to 15 students. And for those of you who don't know, Newcastle is about 2 hours north of Sydney on the eastern side of Australia. And so we have worked over the years in terms of developing all aspects of the technology pathway. So all the way from synthesizing the ink materials themselves at scale, to roll manufacture, to, in the case of printed solar, actually doing installations. I actually joke that I have the highest qualified tradie workforce in the country, PhDs with their working at heights tickets and high-vis jackets and so on, but also printed sensors. And another aspect of what we do is atom beam microscopy, which I won't talk about today. So what do we do and how do we do it? Well, one of the things that we developed over the years is this capability and this facility now to print roll-to-roll organic electronic materials at scale. And this has been made possible through funding by the Australian federal government through an organisation known as the Australian National Fabrication Facility, which is actually quite unique in a global sense, whereby the government funds facilities, equipment, and people to manage the equipment so that it's accessible by all, whether that's Australian or international people. And we have focused on large-scale demonstrations of the technology. And currently we can print at a scale of typically hundreds of square meters of material at a time, whether that's solar cells or transistors, actually. And our next scale up is targeting thousands of square meters. That's quite a lot of stuff. So the challenge for us, of course, is looking at one of the biggest challenges in the world, and that's diabetes. And we already have 540 million people with diabetes. Indeed, this number keeps going up. I've talked about this for, perhaps over a decade. And I just keep having to change this slide because the number keeps getting bigger. And of course, if you have diabetes, then what you have to do, you have to stab yourself 4 to 10 times a day just to work out whether you can eat or not eat or whatever. So our vision way back in 2005 was, what about if you did this instead? What about if you could use your saliva to detect glucose in your blood? So one of the things that is known is that the saliva in your glucose is there. And obviously, blood glucose testing is a major barrier to effective healthcare. People don't like to do it, so they don't do it, and they don't monitor their blood glucose, and then that's okay because it's a chronic condition and nothing happens for a number of years, and then your leg drops off. So, these sorts of things are important, and it turns out, of course, that your salivary glucose level correlates well with your blood glucose level. It's obviously a lot easier to collect than blood. But the issue is that the glucose in your saliva is at a concentration about 1% the level that it is in your blood. And so if you're going to use it, you're going to have quite a sensitive detection system. So what about transistors then? So what is a transistor and how does it work? Well, a transistor is an electronic device that is both a switch and an amplifier. In the sort of electronic logic circuits that we're familiar with, of course, we use it primarily as a switch. It's either off or on, and that gives us binary logic, which gives us all of the, information technology revolution that we've come to become accustomed to, and TikTok and all of those sorts of things. But a transistor is a device that consists of 3 terminals. And I've shown a picture up here. You have a drain, a source, and a gate. And essentially, what you do is you use the voltage on the gate to allow current to flow from source to drain, just like you would have a gate allowing things to pass through. And I changed the voltage on the gate, and that makes really quite big differences to the current that flows. In other words, it amplifies the signal, because even quite small signals, small voltages on the gate can make big swings to the current. And I've showed a graph here which shows that drain current, the current flowing from source to drain. And what you can see is that if I pick a particular voltage between the two, so that sets the current that flows between them. Now, if I change the gate voltage, look what happens. That current increases dramatically as I change the gate voltage. And so we thought, well, that's interesting. What about if we were to stuff a biomolecule in at the gate? The biomolecule respond to some sort of stimulus and could change the voltage on the gate, could change that signal, and therefore provide a big change in signal to the source drain current. The advantage of these organic transistors now is twofold, in the sense that they also have a very low operating voltage, and that's good for biological systems because, of course, they're very sensitive to voltages, and indeed voltages above about 1.5 volts go above what is known as the water window, and things start to break down. And so we've got an electronic system that works at very low voltages. It's based on carbon, and so biocompatibility here is excellent. And so we had the daft idea back in 2005 of, why don't we just stuff the biomolecule into the ink that we print? Well, it turns out that daft idea works. And of course, the first biomolecule that we used was an enzyme, glucose oxidase. And we were able to create really quite sensitive transistors. And so this is a picture of the typical structure that we build and have continued to build. And so we have our electrodes here on our electronic device. We've got our semiconductor, and then we have a material that's mixed in with our biomolecule, in this case an enzyme. We put a droplet of glucose on the top and we measure the response. And so over the years, what we've worked out is that what happens is that glucose diffuses through to the glucose oxidase, and that's embedded simply in a material that conducts charges very well, an organic material, and that's known as Nafion. That's a very standard material. And then that process, of course, produces hydrogen peroxide. That's how the enzyme works. And that hydrogen peroxide, if we get the voltage right between the gate and source and drain, produces protons. It turns it into hydrogen ions. And those protons diffuse down to our semiconducting layer and change its conductivity. And that makes a big difference to the signal that we measure. And so we were able to show that we get sensitivities all the way down to the micromolar regime required for detecting glucose in your saliva. And so I thought I'd show you how we make these things if this is of any interest, and we'll see if the video works. Let's see how that goes, and perhaps someone could give me a thumbs up if this is running. Excellent. So here we are. You have to give the pastiche music. And so we're showing you the roll-to-roll printing machine that we use. And here are the sort of first set of the layers coming out. These are the electrodes. So you'll see, if you look carefully, there are 3 little stripes on each device. And those are the source, the drain, and the gate electrode. And then what we do is we put the semiconducting layer on, and that's done using something known as slot die printing. And here, they're just stripes. We just print stripes. And you can see that coming out here and rolling up. There's the slot die head coming down. And you can see that it forms a little meniscus under each track. And as the roll drags under that meniscus, that's how we get the printed layer first. Now, the printed layer is about 100 nanometers thick, so it's really quite thin. And so you can see those stripes forming there. And this machine, that machine that you saw before, you'd buy it to make wine labels, actually. Now we do some screen printing, sort of stuff you've probably done on t-shirts. And what we're doing now is we're just printing the glucose oxidase and Nafion layers on the top. And so there's the screen printing coming across. And you get another view of it here. And there you are, sensors formed. Tens of thousands of sensors. That's quite enough of that music. So tens of thousands of sensors formed. I mean, typically the roll we print on PET, just a roll of the standard plastic you'd use to make drink bottles. It's about 75 microns thick. We print typically perhaps 0.5 kilometer to 1 kilometer of sensors at a time. And so you can see this is really quite a scalable way of developing biosensors. We haven't just looked at sensors for glucose. What we've shown is that this technology appears to be a bit of a platform technology. I've yet to find a biomolecule we can't integrate into these transistors, actually. And so we've looked at peptides and antibodies with US Air Force research, and we've shown that we can integrate those into the device and produce sensors that can detect the stress hormones in your sweat. We've worked with Pepsi research, looking at producing hydration sensors. So integrating a set of molecules known as crown ethers. They're kind of funky. They have a little ring in them, and the size of the ring you can tune so that a particular ion, for example, sodium or calcium or chlorine, fits in there and is locked in that molecule. When that happens, you get a change in the electrical behavior of that material, And we were able to detect the electrolytes in your sweat down at levels to show whether you're hydrated or not. Here we were able to actually build differential sensors to look at measuring both a reference and an analyte compared to each other. So that was kind of fun. Then more recently, since 2016, we have started commercializing the glucose side of this sensor. This technology listed on the NASDAQ in 2020, late 2020. And we've also been working with the Weiss Institute at Harvard on sensors for COVID, which has been quite successful as well, integrating that technology. Then in 2021, we received federal government funding to build the first factory to make these sensors here in Newcastle, and actually on campus is where it'll be. Developing a new diagnostic test. And so we're now in the process of specifying the buildings, the equipment. The buildings have gone for DA approval. And so that's all in train. So I think I've bored you enough. I'll stop there. What have I told you? I've told you that organic electronics is based around a remarkable set of materials known as semiconducting polymers. These materials can be printed, manufactured across vast areas to produce all sorts of electronic devices. We've worked on biosensors for this talk, but we've also developed printed solar technology. The center at Newcastle is quite broad-ranged. I run a center where I'm a physicist, but I'm in a minority. We have chemists, engineers, biochemists, and so on, all working within the center. So it's a lovely place to work. The technology that we've talked about is a platform technology where we can integrate all sorts of biomolecules into a printed transistor circuit that we can now scale up and produce at low cost. We're currently working on sensors, obviously, for glucose, but also proteins, antibodies, and electrolytes. And our next generation targets are to look at biomarkers for cancer, hormones, and allergens. And with that, I'll stop. Thank you very much.
Thank you so much. And again, if anyone has any questions, feel free to either type them in the chat, or we can go ahead and unmute you, and we will have additional time for questions after Dr. Carlow presents.
Hi, Paul. That was a great presentation. I also love your very highly trained, the highly trained workforce you have there. I was just curious, we've been talking a little bit about diabetes and detecting diabetes in saliva, or detecting salivary glucose, actually just over the last couple of days for a different kind of research project. And so I've got a couple of questions, and that's, if we have stored raw saliva samples that have been frozen, can your sensors be applied on those samples? And also recognizing that, we've been looking into some of the research on measuring salivary glucose, and a lot of the research that's out there suggests, it does have to be fasting glucose, but, recognizing that you've developed a much more sensitive method, what are your recommendations for applications of this?
Okay, let's see if I can tackle both of those. In terms of frozen samples, that would be really interesting. I don't see any reason why you can't. I mean, glucose is a fairly stable molecule. Obviously, I'd have to defrost them first, he said, jokingly. But yeah, I don't see why not. And indeed, we're starting. And in fact, we know that we can do that, because we've been working with colleagues on the west coast of the US, actually in the Bay Area, looking at correlation studies between blood glucose and salivary glucose. And that's a good segue to the second part of your question, which is really, how must you go about sampling the saliva? How do you manage the patients? And here, I think we're starting to learn an awful lot. There have been a number of studies. The methodologies are not necessarily super consistent between them. And I think that we certainly do have to have a standardized protocol. And yes, I do think that's going to involve some fasting. I think it's going to involve some rinsing and making sure that you've got rid of static glucose in your mouth, and then allow that to develop. It looks like passive collection, so passive drawl is probably the best way to collect. But it's still an open question, Anne. I think we're still developing that side of things, the actual sample collection part.
Yeah, well, that's a bit interesting. I mean, we've got these samples, they are longitudinally collected. So they are 6 samples taken 2 weeks apart from individuals. So while we don't have the data on, you know, whether they're fasting or not, even, I don't know if we've got the data on what time of day that they were collected, but, you know, what we're thinking is, you know, if you have an individual who's diabetic, do they just have higher levels of salivary glucose around the clock kind of thing that you can detect and compare? Although, of course, some have it, you know, that, you know, spike up and down. But just sort of in these samples, like, you know, can we, I think our major goal for this is to measure glucose in these samples, because we're seeing a potential association with one of the bacteria that we work with.
Yeah, I mean, you certainly could, and indeed, you could do that. Obviously, both salivary glucose and blood glucose. You could do that comparison. The question would be whether the patient had, for example, had some sort of a sugary drink or something before they did the test, or were they fasting, or whatever. In other words, that may alter the sort of correlation, obviously, between salivary glucose and blood glucose.
So maybe I'll go next. Paul, thank you. Really fascinating talk. Great work that you're doing. Your talk kind of prompted me with a TB question. One of the molecules that is the hottest diagnostic for non-sputum diagnostics for TB is lipoarabinomannan, which is it's a glycolipid and a virulence factor that's found in the wall of MTB. And actually, the major barrier to scaling it up as a test, there's a commercially available test that's used on urine, but it just lacks appropriate analytic sensitivity, except in certain individuals, such as people with advanced HIV. And so I'm wondering if your platform could detect a glycolipid, would that work? And have you done any studies in urine?
The answer is I don't know, and no, I haven't done anything in urine yet. The interesting thing would be whether we could get a particular species that would bind to that glycolipid. It was very interesting you should say that, because during your talk, I started searching for what the biomarkers were for TB, because I thought, oh, this is interesting. I wonder if we can do that, and indeed picked up that glycolipid. So I think the question would be, I don't know the chemistry of that glycolipid and whether there are already tests that bind that glycolipid or work on locking into a particular aspect of that molecule. Because if we can do that, then I think the answer is probably yes, because you could then bind to the gate, and then we will see a change in the field that's generated.
Fascinating. Thanks, Paul.
It's a great question.
All right. Well, thank you all so much. And we will have time for additional questions at the end. But now I would like to invite Dr. Carlow to present.
Thank you to Brittany, Samantha, and Anne for the invitation to speak with such an esteemed group today, and for the opportunity to hear 2 amazing talks. So a little bit of a hard act to follow here, but I'll do my best. So what I'm going to talk to you a bit about today is some of the infectious disease research that's going on at NEB, New England Biolabs, and specifically in my lab, and how we have focused on developing some tools for diagnostics that we have applied in our CLIA lab here at NEB for COVID testing, as well as some tests which have been applied elsewhere in the world, specifically the tropics. So first of all, a little bit about NEB for those of you who don't know. So we were established in 1974. We're located in Ipswich, Massachusetts, just a little bit north of Boston. And we produce and supply a whole range of reagents that are used for both life science research as well as biomedical applications. We have a GMP manufacturing facility so that a lot of our reagents can can and actually are used in all kind of medical settings and diagnostics. Our Chief Scientific Officer is Dr. Rich Roberts, who is actually a Nobel laureate, won a prize for looking at the actual discovery of introns in eukaryotic DNA, as well as the mechanism of gene splicing. And we have a large research department where we actually conduct basic and applied research in all kinds of areas, including infectious diseases, which is really the focus of my lab. So one of the areas that we have worked on for actually almost 4 decades, 40 years, is focusing on neglected tropical diseases. The idea here is more of a humanitarian effort where we hope that our basic research can be used to help to control some of these really nasty diseases. And what I'm showing you here are actually 2 diseases which are caused by parasitic worms. These are nematodes, and they are transmitted by different types of insects. So they're particularly nasty diseases. And what we are, one of our goals is to actually come up with new types of tests that can actually be used in low resource settings to detect infection in both humans as well as the insect vectors. So traditionally, these diseases are basically, it's a microscopy method where you're taking a piece of skin or a piece of blood or an insect, and you're basically looking under a microscope for the presence of parasites. These methods are have a very low sensitivity, fairly difficult to do, in fact. So what we're hoping to do is replace these methods with more sophisticated, more sensitive methods that can actually be used, though, in low resource settings. So one of the methodologies that we've spent quite a bit of time focusing on is something called LAMP, or loop amplification, isothermal amplification. And one of the nice things about this method is it has a whole variety of different readouts, and one of them is a colorimetric readout. So this is something that's really simple to read. We're basically looking for a color change from a pink color to a yellow color if the sample is positive for whatever pathogen we're actually looking at. And we've developed a whole suite of these tests for these neglected tropical diseases, the filarial diseases. And these have been used for surveillance in places like Cameroon, Brazil, Colombia, and Venezuela. And we've actually published a number of papers where we've actually discovered biomarkers and then developed these very simple colorimetric tests for use in the field. We've also been focusing on some diseases that we find both worldwide as well as locally here in Massachusetts, and these are tick-borne diseases. So we've also developed these simple colorimetric tests to look for a variety of pathogens in ticks, which are really important vectors of diseases. And I've listed here 3 of the diseases that we have developed tests for. And these are, I'm showing you some of the ticks that have actually just been collected from outside the lab here. And they do contain quite a few of these pathogens. Speaking of other vector-borne diseases, Triple E that's Eastern and equine encephalitis virus, is also a very important disease here in the United States, transmitted by mosquitoes, normally cycles between birds and mosquitoes. But we do have situations where the virus does get into horses and people. So that's something that we were working on fairly recently, which is the identification of new biomarkers for this virus, as well as developing LAMP assays to detect these viruses in insects as well as in mammals. So this was work that was done in collaboration with Professor Unash at the University of South Florida, where he was able to actually validate the various tests that we developed here and showed that they can actually be deployed for mosquito control programs. So switching gears a little bit again, talking about coronavirus. This is something obviously that's very much in everybody's mind still today. And obviously it's a virus that's normally found in bats. And in 2019, the whole world basically turned upside down when we were experiencing the start of a pandemic. So back in March 2020, we decided to see if we could actually develop a LAMP test, something that could be deployed both here and elsewhere, something that would be, again, simple and suitable for low resource settings. So at that point in time, it was quite hard. Most of the testing was actually done with nasopharyngeal swabs. We opted to focus on saliva, which is obviously very much of interest to this group here. And for us, it was a bit of a learning curve because we had worked with blood, skin, and a whole variety of insects, but this was a first for us to actually work with human saliva. So as I mentioned, what we were hoping to do was to develop a simple colorimetric test that did not involve a swab, and that it was something that we could directly use with just raw saliva, hoping to circumvent the use of an actual RNA extraction step. So fortunately, our colleagues here at NEB had put together a really nice kit containing primers and a master mix that could be applied to all kinds of different situations. So that was basically the kit that we used. But as I said, for us, there was a learning curve. We had to learn how to work with saliva. And we learned that it can be quite a variable sample to work with. I mean, I'm showing you here just 5 salivas collected from the same individual at different times. It can be different colors, different textures. And for our test, it's actually quite sensitive to pH because the color reaction is actually a pH, basically it's a pH based reaction. So we learned that certain salivas were very acidic that could interfere with our test. We started by collecting saliva from volunteers here at NEB through an IRB approved process. We tried a whole bunch of different viruses spiked into saliva to see how that could actually impact the detection. And we tried a whole bunch of different ways to try to break open the virus to release the RNA so that we could detect it, preserve it, and do this all very rapidly. So here what I'm showing in the white box is the actual buffer that we settled on, which is very similar to a buffer that was described by SEMCO group. We added a detergent there because we found that actually was rather helpful. And from there, we basically scaled up and started the process of actually trying to, or hoping to actually implement this protocol and this method for testing our employees here at NEB. So in order to do that, we had to establish what's called a CLIA laboratory here in the United States. And we had to submit our tests as well as data to basically demonstrate that it adhered to a standard that was required for testing of patients, showing that we had reproducibility and reliability every time we actually performed a test, regardless of who was performing the tests. So we did get this certification, and in April 9th, last year, we actually started testing our employees. So this is kind of a workflow that we use here. People basically spit into a little tube through a pipette tip. They register their sample using a barcode. And the first thing we do is take a small amount of that while we inactivate the sample, the virus in the sample. We take a small amount of the material from the saliva. We add it to the lysis buffer that I just described for 5 minutes, heat at 95 degrees. And then from there, we take a small sample and we add it to a plate that has a master mix for COVID detection. We have another sample that goes into a plate to detect human actin, which is our positive control. And essentially, what we're looking for here is a colorimetric readout, or in the case of our CLIA lab, we also use fluorescence to give us a quantitative readout. And basically, when the sample turns yellow, it's positive, and that's true for also the actin. And if we actually have a positive COVID result, we actually go ahead and we actually repeat that 3 times before we release that result to the patient. And the test has to be validated by having a positive actin. If the test is invalidated because we are unable to detect actin, we repeat that again 3 times, and we then release that result to the patient that the test is invalid because of the quality of the saliva. So we report these results to the patient, to actually NEB, as well as to the states in which the patients actually reside, and all this data is kind of is collected by the state. So in addition to reporting the positives to our colleagues and patients here, we also sequence every single sample that's positive, and we use both Illumina sequencing and nanopore sequencing here to get that result, to basically identify which variant is present in that saliva sample. So this is just a snapshot of where we are today. As I said, we started back in April 2021. Initially, we were testing people once a week. And then as we started experiencing surges, we started testing twice a week. And now we are actually testing 3 times a week. So we've collected a lot of data, a lot of information, as well as what variants are present in these samples. And basically what we've been doing here is really mirroring what's seen nationwide and actually worldwide. So we have quite a lot of information from a very specific cohort of people. And this is kind of where we are today. Here at NEB, using this saliva test, we've actually processed almost 90,000 saliva samples. I'm kind of showing you here what our positivity rates. We've identified approximately 300 people that have, over the course of this time, tested positive for COVID. Most people have tested, this is about actually 60 to 65% of our study population here, which is around 400 to 500 people testing 3 times a week. So it's quite a surprising positivity rate. Most of the majority of our colleagues here are vaccinated, but even today, as of this week, we're experiencing a little bit of a surge after our Thanksgiving holiday. So we've published this method and made it available to the community. We also shared this protocol, this saliva protocol, with colleagues at the International Center for Genetic Engineering and Biotechnology, and they've actually deployed this test in various places in Africa and published this manuscript here. This is work funded by the Bill and Melinda Gates Foundation. And again, all this is just based on a very simple saliva test with a colorimetric readout based on this LAMP assay. So more recently, though, I'll just talk about it for a few minutes here. We've kind of switched gears a little bit, and we've started working on the development of again, a simple LAMP test to detect monkeypox or mpox. So as you know, there was an outbreak recently, and this is a disease which is normally, self-limiting, not normally fatal, but certainly something that was of concern. So we started by just doing some analysis of the phylogeny of this group of viruses where the monkeypox and the mpox actually resides. These are the orthopox, and we looked at about 100 mpox genomes and about another 100 orthopox genomes, and we looked at how these viruses actually relate to one another, and they're actually extremely closely related, and that actually presented a bit of a challenge when you're looking for a specific test. And they're also, as you know, very like zoonotic, so they jump from one species to another. And in fact, the original smallpox vaccine was probably actually a cowpox or a horsepox rather than vaccinia itself. So where we are today is we have explored these genomes, and we identified 10 different genes which look promising to us in terms of a biomarker for this disease. And we developed 13 different LAMP primer sets targeting these different genes, as well as the gene that's used in the CDC test. This test or this gene is a little bit problematic because there's a lot of mutations which are going on in that gene, which could present a problem again because of dropout of these with respect to diagnostics. So of these 13 primer sets, we settled on one primer set that we thought which is actually very, very good as a sort of pan-specific test for any of these orthopoxes. So whether it's smallpox, cowpox, monkeypox, basically we have a set that works really well for all of those. And then we also have a primer set and a test which involves the use of a probe, so it's not colorimetric readout, that is actually specific for mpox. So this is just kind of showing you a little bit about, this is the pan-specific test, and here I'm looking at camelpox, vaccinia, and monkeypox. Basically, you can see that everything's turning yellow, so we're detecting all those different DNAs. I'm also showing you some fluorescence values here that gives you some kind of sense for titers. And this is, looking at the performance of our specific monkeypox specific test. So it's a LAMP test, but we use a probe. And here you can see the only thing that's actually been amplified, which are the green and the blue lines, are either the monkeypox DNA itself, which is actually really hard to get hold of, and a G block, which is a kind of sort of proxy for this this specific virus. Camelpox and vaccinia, there's a flat line across the bottom. So so basically, where do we go from here? There's been a few papers just even the last couple of weeks showing that there is certainly good levels of monkeypox virus in saliva. And there is a test that's been published already. But what we're hoping is that we can validate this test by receiving some saliva samples and basically demonstrate that this test could also be used for a saliva detection, as well as useful in places where there is really very low resource settings here. So with that, I'd like to end by thanking many people at NEB. There were so many people involved in all different departments to actually develop this COVID test that I described, the kit that we then applied to a saliva-based diagnostic. Colleague at Miramis who provided us with saliva samples. And for the non-COVID stuff, I'd like to call out the people that are actually in my lab and the CLIA lab, as well as our collaborators all over the world who are basically trying to deploy these different lab tests for the detection of different pathogens. So with that, I'd like to end and thank you all for your attention today, and happy to answer any questions you may have.
Thank you so much, Dr. Carlow. If anyone does have any questions for Dr. Carlow, feel free to put them in the chat or raise your hand and we can unmute you.
Dr. Carlow, just thinking about the innovations that you were sharing with NEB and how you guys were required to pivot, curious to know if you've got any thoughts about your ability to take that experience to respond to the pandemic within your office and be able to use that going forward. Is that something your team is exploring and digging into in addition to the science?
Yeah, so I mean, it's become really an important tool for us. We are able to identify people really early in infection, asymptomatic people. So with that, we're able to basically ask them to go home, and oftentimes they're a little bit surprised with the result, and then afterwards, a day or so afterwards, they start feeling really quite poorly. So it's really helped us to keep our population here safe. It was really helpful throughout especially the early stages of the pandemic, where it was really important that we were able to try to ensure that we could continue to supply really the world with these kind of tools, not just for detection of COVID, but as well as vaccines and everything else. So it's been really very important for us, and as I said, being able to share the protocol as well. It definitely kind of fits with our philosophy of especially when it comes to the tropical diseases, of hoping that we can help control the spread of some of these nasty diseases, including COVID and potentially here also monkeypox.
Thank you, appreciate it. And it looks like we have a question that came through in the chat from Orchid about, do you have an intention of developing a RSV test?
It's not something we've started. It's definitely something I could see again. You know, we're always very interested, especially in my lab, about what can be applied, not just, you know, in the United States, but like worldwide, something that was simple and with an easy readout. So that's definitely something that's really worth consideration, especially, you know, where we are today, right now, where we have this kind of triple pandemic with, you know, flu, COVID, as well as RSV in children. So it's definitely a very worthwhile, definitely worthwhile to consider that for sure.
Well, all three of our speakers are still online. So if anyone has any questions for any of our three speakers, feel free to place those in the chat. But thank you again so much. I learned a lot. And I really appreciate all three of you being here. So again, if anyone has any questions, feel free to ask away.
But I just wanted to jump in, especially with my New England Biolabs mug, and my camera has frozen, but with the lab assay right next to me on my desk. That's a common feature for my oatmeal / porridge, whichever you want to call it. So I appreciate that. I really appreciate the work that NEB has done, especially their ethos of, you know, helping research and testing initiatives around the world. But I also just wanted to say thank you to all of our speakers for joining us here today. It is just great to hear. It's, yeah, I learned a lot from each of your different talks. I'm very inspired to see where else this is going to go. I'm very just excited to get so many of us together in one place and to keep these going. And of course, big thank you to Brittany and Sam for organizing this.
I can't think of a better closing than that. Thank you, Anne, for that. Appreciate it. And very, very grateful for our speakers who've all have joined us, given up their time. We had an adventure of figuring out what time zones would work to be able to have this panel pulled together, and very, very appreciate you all joining us.